Okay, we are live. Hello, everyone. Say hello. Hello. You're saying hello to yourself. Okay, great. All right, so what I want to do first is talk about a little bit of communication. We looked at the handout a couple of times now for immunity, and we saw that there's some very clear distinctions between what a complete antigen is and incomplete antigen, what specific or non-specific immunity is, right? Well, this today is specifically going to be, all about the learned immunity. We're going to cover it pretty much in detail. So I want to start by saying a couple of things. The way T cells and B cells behave are going to be a little bit different. So B cells are going to behave under what's called humoral immunity, and T cells are going to be cell-mediated. This is just more like word association, getting some terms straight in your head, okay? With B cells, we're going to see something come out of it called plasma cells. Remember, B cells and T cells are nothing but lymphocytes. So if we want to put that as our heading, going back to the blood chapter so we know where we are, sometimes a little familiar ground can mean a lot when you're looking for something to stick. So we're just talking about our lymphocytes, right? Of these two lymphocytes, the B cells and T cells are both produced where? They're in the bone marrow. They're matured in two different places, right? The B cells stay in the bone marrow. T cells migrate to the thymus under control of a hormone, thymosin, to start maturing and all that kind of thing. Okay, good. Well, under B cells, when they find their antigen, anytime there's that binding of the antigen, right, to the B cell receptor, that's what they call a complex. When that happens, that's when you're going to get these clones. So you're going to get clones formation out of the B cells. Some of those will become plasma cells, and some will become memory cells. So only upon stimulation of an antigen, let's say, do we get these cloning happening on both sides. Both T and B are going to clone themselves. But the words that we're going to use here in B cells is plasma cells. Out of the plasma cells, this is where we're going to get antibody production. So let's just say 80% of those clones, I'm just throwing out a number, I really don't know the exact number to be honest. 80% of these clones are going to become plasma cells that will then secrete antibodies that will match the original antibody that happened to bind with the antigen, right? The immunoglobulin receptor, we'll see that in the video. And the other 20% of the clones will become what? Memory cells, which are going to be tucked away for later, right? They're going to be saved for a second exposure. Are we all good so far? Okay, good. And that's occurring all around the bloodstream, right? Which is why they call it humoral immunity. All within the bloodstream here. Whereas T cells, they police the actual individual cells, right? They're not necessarily in the blood. They are in the cell space, the interstitial space, looking at cells specifically and act directly upon cells. So we call it cell-mediated. In this part, T cells, we have two main kinds. We'll have a T helper, and we'll have a T with a C next to it, which we call a T cytotoxic. So for cell-mediated immunity, we're dealing with two main types of cells. I know there's a little bit more than this, but we're going to keep it simple. A T helper cell and a T cytotoxic. Well, think of this as two different T cells that speak two different languages or have two different handshakes. The the T cell that's the cytotoxic kind, this is going to have the CD8 receptor. And the T helper is going to have the CD4 receptor. Are we still good? So we have two different receptors, and guess what? That means that they're going to be specific, or they're going to want to handshake or communicate with two different kinds of cells, since they have different kind of receptor, right? The cytotoxic T cell is the more dangerous kind. This is the one that has the poisons that can kill cells right out, just like we saw with the natural killer cell. Same kind of mechanism. The T cell can come right up to the infected cell, inject some poisons, and the cell lyses. So it's a nice quick story for the cytotoxic kind. This right here is going to be specific for linking up with our B cells over here. So it's the helper cell, which is why it's called the helper, that will help manage a very large scale response and will call upon many more antibodies to come to the area. Right? It's like a general. This one's like special forces, the cytotoxic, where it cell-to-cell connection, it just kills it right outright. Whereas the helper is going to want to bind to our antigen-presenting cells and our B cells. When it binds to that, it learns from it and then creates more clones and then mounts more an antibody response. So this is more complicated, but it's necessary, right? This is more straight out and also very necessary. Here's the deal. We mentioned so many words in our handout. This is how you link it up. This T cytotoxic cell, which has the CD8 receptor, will have the ability to recognize what's called class 1 MHCs. So now we're associating another word, another term. Cytotoxic T cells will have class 1 MHCs. Right? They'll recognize class 1 MHCs on the tissue cell, rather. Whereas the T helpers, they're going to have recognition for class 2 MHCs.
What does MHC stand for?
What's that?
What does MHC stand for?
MHC stands for major histocompatibility. And we're going to see it in the PowerPoint. I'm just giving you some terms that we're going to be able to shoot back to throughout the lecture today. Because this is what you need to put together. It is the hardest part of the chapter. And basically it's just trying to get the terms straight in your head. What does what, okay? So what we've said was now B cells give us our plasma cells and memory cells. Most of these will become plasma cells of the clones, which will produce our antibodies. What do the memory cells do? They tuck themselves away in all of our secondary lymphatic organs, things like the lymph nodes, the spleen, all that kind of thing. They just sit waiting for a future exposure to that same antigen. They're all primed and ready to go. And we'll see that, how they affect on the second exposure. Now on the cell-mediated side, we have two main types of T cells, the cytotoxic and the helper. The helper is going to have the CD4 receptor and it's going to recognize what? Class 2 MHCs. The cytotoxic one will have the CD8 receptor and will recognize class 1 MHCs. The last little bit we need to say is that where in the world are class 1 MHCs?
All over the body.
These are all on our tissue cells. So all of our cells will have on their membrane, right, we talked about cell membrane way back, AP1. On their membrane, on their cell membrane, they'll have a certain protein marker, right, called a class 1 MHC. You get that? Okay. And on the other side, T helper, it will want to communicate with class 2 MHCs. Where are these? These are special protein markers on not all body cells, but only on certain cells like B cells and APCs, antigen-presenting cells. Which is why now, if you want to follow me with this crazy arrow, we're going to look at the class 2 MHCs over here and know that we're going to link that up. Right? With, whoops, let me carry the arrow over here. Link it up with our B cells. So guess what? Now we know that when a B cell ingests an antigen, what's it going to do? It has the ability to digest that antigen, break that antigen into protein fragments, use one of those fragments and stick it on its membrane surface like a flag, saying, look at me, I'm infected, or I've ingested something that could infect me. Now it's going to call upon what? A T helper cell to bind to that with its CD4 receptor. Remember, the class 2 MHC is over here on the B cell. The T helper scans our tissues, looks for infected or, bound to antigen-antibody complexes, okay? So that's what we're going to see in the PowerPoint. That's just a little appetizer.
Basically, the B cell marks itself as infected.
Yeah.
Digestion.
Yeah, well, I shouldn't, it's my mistake, I shouldn't use the word infected. I should use the term, the fact that it's bound to an antigen. It may or may not be infected, okay? Because we're talking about a B cell, we're not talking about a tissue cell. The B cell necessarily just absorbs the antigen, right? Yeah. And then carries the fragment of the antigen to the cell surface. And that's going to be the beauty of these videos that we're going to see. Okay. All right, so, I'm not exactly sure where we left off last time, but
We made it to that slide, basically.
yeah, that's what I thought. Okay, so just as a quick review, because we really do need to smoke this today. I think we can. Because that right there is really helpful if you can understand what's going on with that and then watch the videos, which we're going to do. Bring it together. Okay? So if we can get all this straight, everybody good on this? What's the difference in innate versus adaptive or learned immunity? We're born with all these qualifications here, right? We don't need to really learn that. We're always going to have inflammation. We're always going to have natural killer cells and fever and phagocytes circulating around. That's innate immunity. So that's first and second line. The first line specifically being our membrane, right? Our membranes, the cutaneous membrane on the outside of the skin. So in the adaptive, what we're going to talk about today is our learned third line of defense. Okay, so I can skip over some of this here. If we want to review the steps of phagocytosis, what